Tēnā koutou katoa, ko Ingarani me Piripini te whakapaparanga mai, ko Leicester me Bristol te whenua tupu, ko Tamaki Makaurau te kāinga, he Industry Research Liaison Manager o i New Zealand Food Safety Science and Research Centre, ko Kyla Archer toku ingoa, tēnā koutou katoa. On behalf of the New Zealand Food Safety Science and Research Centre, I'd like to welcome you all to the July edition of the Food Science Seminar Spotlight of 2023. Firstly, thanks to those of you who were able to join us in person at our annual symposium at the beginning of July. Hopefully you weren't too hampered by the weather or flight issues. For those of you that were and weren't actually able to meet with us in person, or if you want to relive your favourite session, these will be online in the next couple of weeks. And the entertaining Kim Hill panel is already live on the Radio New Zealand website. You can find the link via our website, and you've also received some emails about this too. So today I'd like to introduce our speaker. Eric Alterman is an adjunct associate professor at the School of Veterinary Science at Massey University. As an independent science, he is also the director of Blue Barn Life Sciences Limited and chief science officer at PPDX in Oregon, USA. He received his PhD in microbiology at the University of Kaiserslautern, Germany in 1999. Between 2000 and 2006, he worked at the North Carolina State University, developing bioinformatic tools for microbial genomics. He was involved in the first lactobacillus genome sequencing project and part of the lactic acid bacteria genome consortium, and the first large-scale microbial genome sequencing project conducted by the Joint Genome Institute in California. Between 2006 and 2021, he held positions as senior research scientist and principal scientist at Ag Research here in New Zealand. And then together with distinguished Professor Nigel French at Massey, he is co-leading the MB Endeavour programme Smart Bioplastics and explores the commercial application space through the Centre for Bioparticle Applications. So as always, if you have any questions that crop up during Eric's talk, please pop them into the Q&A at the bottom of the Zoom window, and I'll put your questions to Eric after the session. So without further ado, I'll hand over to you, Eric.
Thanks, Kyla. That was a very kind introduction, and thanks for everybody who's joining in this webinar. We've practiced this before, so I'll try to share the right screen. Please, we should be able to see what I'm seeing now. If not, Kyla, please yell out.
All good.
Excellent. So the title of the talk is Smart Bioplastics, Pursuing Sustainable Food Packaging. And before we go further into the talk proper, I would like to start with a small plug for our new Center for Bioparticle Applications that really tries to unify all the activities that are happening and will be happening in this space, particularly around research and then translating research into commercial applications. So if you find the topic, the content of this webinar interesting, please feel free to jot down the URL www.bioparticles.org and have a look through the videos and the individual programs that are shown there already. All right. So when we started a couple of years of putting this program together, there were actually two things that really struck us. One was the amount of food waste that is thrown out on a daily basis for various reasons. And the other one is an equal staggering amount of plastic waste, partially from food packaging, but also others, that end up in landfills and then ultimately make its way into the wider environment and start polluting everything. And if we look at this a bit more detail, then we can see that there's a lot of food waste actually really happening, particularly with fresh foods. If you look at fruits, vegetables, various types of fish and beef and meats, we can see that often less than half the amount of food actually produced is consumed. So more than half is lost and thrown out either at the consumer level, or the retail level. And to me, that is an absolutely staggering amount of waste. And in part, this waste comes through limitations in shelf life. So if you look here, say for spoilage organisms if the amount of spoilage organism reaches a certain threshold, or the amount of food pathogens on a particular food item reaches a threshold, then the item is either not safe or less or not desirable anymore for human consumption, is being thrown out. So if you can lower and flatten those curves, we can extend shelf life and help contributing to having less food being thrown out and being wasted. And then there's the other aspect around an ever increasing plastic accumulation in the environment. And if you see here, the amount of plastic going into the waste management system from 1960 all the way to 2018, overall, there's a staggering amount of plastic being produced. And unfortunately, even since as early as the 1990s, attempts of doing something with this plastic other than putting to landfill are very limited. So we have here in yellow combustion with energy recovery, and there's hardly any growth. It's very static. And then also in the early 1990s, recycling comes along, and even that really doesn't go anywhere. So it's all very static. And the only bar that really grows is the amount of plastic that ends up in the landfill. Now, if you look really carefully, there's also an item in here that says composted. And I left it in because it was actually in the original data set, but the numbers for composting remained 0 all throughout. So the amount of composting that's actually happening in terms of plastic reuse is so small it doesn't even register on the scale. But on the other hand, I also believe that actually provides an opportunity for all of us to say, can we actually do something about creating materials that can be composted and start chipping away at this really large blue bar that goes into the landfill. And things have happened. There are materials out there that are bioplastics that can be composted. And the reason, or one of the reasons why there's so little of this still on the market, comes down to simple economics. So if you look here, for example, at the cost of petroleum-based plastics, just a simple one, PET, and we see over a year and a half, roughly, we see that the prices for virgin PET flakes is actually dropping from about $1,300 per metric ton down to less than 1,000. If we compare this to, say, food grade pellets, they initially follow this trend and then stabilize. So let's say they're sitting around $1,500 per metric ton. So keep that number in mind. So this is for regular petroleum-based package material that you use for food packaging. Now, if we go out and want to do something for the environment, we want to find bioplastics that are either made from sustainable materials and ideally can be recycled or biodegraded and composted. And I went on some large Chinese websites because in all reality, that's where most of the stuff comes from for reasonably cheap. And the cheapest one I could find after some searching was for PLA, which is polylactic acid, food grade PLA granules, selling at the moment they are between $4,500 to $4,700 per metric ton. Depending on the actual material you need, this price can go up significantly. And this means that for the same functionality, food packaging, you have to suddenly look at a price increase of 2.5 to 10 times against the petroleum-based equivalents without any other benefit to it. And it's very hard, I believe, for any manufacturer to justify this cost if all you get back is essentially a marketing opportunity to the consumer. And one of the things that has been tried in the past and is being tried is to actually overcome this price differential by adding value to those materials, for example, by adding antimicrobial properties that extend shelf life. So if we can flatten the curves, remember from the extending shelf life, and extend it by days or weeks, then that would be a real value add that would possibly justify the higher cost for this material. And early this year, Andrew Manley from the Active and Intelligent Packaging Industry Association has written a very interesting commentary in Packaging Europe. And he actually makes the observation that there are lots of innovative antimicrobial materials that are produced in research labs across the world that are really effective and do work, but hardly any of those actually reach scale or come to market. And that is a huge gap. So there's lots of stuff that's out there, hardly anything is being used. And so he finishes up in saying, what really is needed are effective and scalable materials or additives that are affordable, cost-effective, and meet sustainability criteria. So these are the two key aspects that we have to hit if we want to have bioplastic materials that are compostable or biodegradable become a real reality for food packaging. And so, finishing up with this part of the webinar, I'll just summarize some of the key findings that we found. So one of the really important parts that industry has approached us and is looking for is extending shelf life, particularly for fresh food products. Along with that comes increased food safety and consumer safety, particularly if you're looking at limiting or inhibiting the growth of food pathogens or spoilage organisms on food items. But a really big item also is sustainability. So it becomes an increasingly obvious that we can't really continue the way we have worked over the last 40 years. And so new materials that are created from renewable sources have to become a reality. And so that also then follows into to limit or reduce the use of petroleum-based packaging materials. But also, and I haven't really touched on this part yet too much, is to reduce the complexity of existing packaging materials. So a lot of the modern packaging materials are actually comprised of 5, 7, 8, 9 different layers of different plastics and sometimes even metals that are tightly bonded together. Trying to unbond those layers and make them and feed them back into the recycling or biodegradation route is almost impossible. And so if you can find or develop materials that help reducing the complexity of those layered materials, it could also help in making them much more amenable to biodegradation and recycling. And finally, none of this really matters if there is no translation of research to commercial application. So this down here, this last point is probably ultimately the most important one, to make sure what comes out of the research lab can be scaled, is affordable, makes commercial sense to apply to then actually provide the incentive. And so that really gives me a nice segue into the Smart Bioplastics program, because we are setting out to address at least some of those research gaps. So one of the main aims of the Smart Bioplastics program is to develop novel biodegradable and or edible food packaging materials that extend the shelf life of fresh foods. And our model system for the Smart Bioplastic programs is currently meat. So we're looking at Clostridia and Campylobacter as our model organisms. We also have trying to make sure that the life cycle of our new materials is much more favorable than the ones for petroleum-based plastics, to make sure that in the end we're not ending up with material that is actually worse than what we have now. That wouldn't make much sense. And finally, and this is pretty much a new research aim or goal that has been brought to us by our industry reference group, again, as mentioned, reduction of the complexity of food packaging materials. And how we're going to go about this is in the next slide. It's a bit busy, but if you bear with me, I'll walk you through this. So this is essentially a very high level overview of what we are doing and who's doing what in the Smart Bioplastics program. And I hope you can all see my little laser pointer here. And so I work essentially a little bit backwards through this program. So the ultimate outcome of the Smart Bioplastics program is to develop food packaging materials down here. And we have our material sciences teams from Sion here in New Zealand and Oregon State University that are specifically looking at these research arms. Now, Sion is tasked with functionalizing existing bioplastic films. They're provided by Convex here in New Zealand, and investigate and develop methods to functionalize them based on water-based coatings and thin film formulations. And so we spray something on there that sticks to them, and then, for example, shrink wrap those new materials onto existing simple food shapes, and these can be part of traditional layered packaging materials. And then the direct contact between those materials and the food item provides the antibacterial activity. Our partners at Oregon State University look at something quite different, and they are experts in developing edible films and coatings. And so these edible materials are made from pumice, from grapes, apples, and pears, for example. And here the idea is that, and that's currently one of the research priorities for this team, is to develop casted edible films that are multi-layered then with to be functionalized. And again, same thing, those films could then be used to contact wrap simple food shapes, or they could also become part of traditional layered food packaging materials with the aim to reduce the complexity. And perhaps the biggest stretch goal for our program is to eventually develop edible bioplastic coatings, so liquids that are functionalized, where more complex food shapes, such as, say, a whole chicken carcass, fruit, vegetables, can be immersed into a liquid solution that then hardens. And this edible coating would then become part of the meal. You would eat it together with the food item, so there would be no food waste whatsoever left. And those novel materials would then be subjected to our quality control, which looks at material properties. So do those new materials and films still meet industry needs and requirements for food packaging? Are they actually compostable? So and what happens during the composting? So make sure there are no secondary biotransformations happening that create anything toxic, or that they're actually not composting at all. And finally, we're also looking at life cycle analyses throughout the entire manufacturing and end of life cycle to again make sure that what we develop is significantly better than what we have right now. And the final part, and that I'm coming now slowly to, is which I'm sort of skirting around for a bit, is the mystery ingredient that we use for actually functionalizing all those different materials. And these are called bioparticles. They are small bioplastic particles between 250 and 300 nanometers in size that are made inside bacterial cells, or microbial cells rather, and they are naturally occurring carbon storage particles. And so they're widely spread in nature. And what we are currently doing, and that's the microbiology and biotechnology teams, we are looking at trying to develop methods to scale the production. So the team in the early 2000s who has actually developed the system left it at lab scale. Again, we come back to what Anne-Romaine was saying from the Packaging Association. Lots of things are really cool and developed at lab scale and then never make it to commercialization. And so the Method team here is really looking at developing a completely new genetic background for the scalable production in large bioreactors for those bioparticles. And I will discuss and explain a little bit more what's behind those and how they work in a couple of slides. But then we also look at a subsequent harvesting and purification of those bioparticles. One of the core things that can't be talked away, and that is simply part of the whole technology, is that it's based on genetically modified organisms. no way around it. But the final particles are just recombinant protein and no live cells are present. So we're going from GMO to recombinant protein, and in New Zealand, there are well over 70 recombinant proteins already approved and in use for human consumption in the food chain. So while it is GMO, it is not something terribly new to introduce to New Zealand and internationally as well. And finally, the Ag Research team is looking at food safety. So they are making sure that whatever bioparticles that are functionalized we create are actually active against the food pathogens and spoilage organisms that we test here, because there's no sense in having the material science teams working with inert particles that don't have any function. And so let's go a bit more into detail on the science a bit, and let me explain to you how those bioparticles actually work and what they are. So here is a quick slide with a little bit of biochemistry and a little bit of genetics, and I'll hopefully can bring this over reasonably easy. So the whole system to make those functionalized bioparticles currently comprises 3 genes. 2 genes here are produce helper enzymes that take metabolites out of the central energy metabolism and activate them and prepare them for the use of the 3rd enzyme that then takes those activated precursors and synthesizes them into long biopolymer strands, and they are called essentially polyhydroxybutyrate. That enzyme as a dimer stays covalently bound to its own biopolymer, and as these accumulate in the cell, because a biopolymer doesn't like water and tends to self-aggregate, once a certain concentration is reached, then they spontaneously aggregate into small spheres, and which we call bioparticles. So that happens naturally. What we can do now is to go in with genetic tools and create gene fusions. So we can merge the different genes to the gene that then makes the enzyme to make the bioparticles. And what this means is that now that we have bioparticles with the enzyme that makes the particles sticking out on the surface, if we have protein fusions, then those proteins are now directionally displayed, covalently bound to the surface of those bioparticles. So now we have function, which means we can functionalize those naturally occurring particles with any enzyme or protein that we want. So that opens up a huge range of different applications for us. And I'll come to that at the very end of the talk. And how do they look like? And what we can see here on the right is currently our production host, E. coli, and we are producing in this picture bioparticles that display green fluorescent protein. So they essentially start glowing if you expose them to UV light. And each green dot here represents an entire E. coli cell, and you can see those cells are pretty much packed up to the limit with bioparticles. So literature shows that up to 8% of the bacterial cell mass, dry cell mass, can be made out of those bioparticles. And then here on the right side is an electron micrograph of purified bioparticles, and you can see they're going from 10 micrometer to 500 nanometers. And so these bioparticles are significantly smaller than bacterial cells. And on the surface of those bioparticles, if you trust me enough, sits the green fluorescent protein being displayed. To activate them actually, and to make them useful for extending shelf life, you need to display enzymes on there that inhibit bacteria and microbes. And one of the most effective enzymes for killing bacteria comes from bacteriophage, so viruses that only attack bacteria. And this here on the left-hand side, I'll just quickly run you through the life cycle of a phage. So the phage attaches to the cell, injects its DNA. The DNA then essentially hijacks the machinery, the cell machinery within the host cell, and forces it to replicate and make new phage. And other enzymes. And one of those enzymes is a Pac-Man here, which is called an endolysin. And at the right time, the endolysin traverses to the cell wall and hydrolyzes, dissolves the cell wall. And then the cell explodes and releases new phage viruses that then can go back up here and repeat the cycle. This method has been used quite widely in medical applications. So back in Russia in the early 1900s, and now modern times also coming into the Western medicine, is the approach called phage therapy. So you use entire phage to inhibit specific bacteria, in this case, pathogenic bacteria, which is very helpful and useful, particularly now with emerging antibiotic resistances to many human pathogens. The question then now, of course, comes, well, if this works so great, why don't you use just phage and for the functionalized packaging? And why do you go through the whole hassle and putting individual enzymes onto the bioparticles? That seems quite unnecessary. Well, the problem with phage therapy is that phage are extremely specific. They're often strain specific, and bacteria are not defenseless. So there are a multitude of phage defense mechanisms within bacterial cells that are highly adaptive, which means that if you have a bacteriophage that attacks a given strain, within a couple of generations, there will be resistant bacteria emerging where the phage becomes entirely inactive. And so you have this constant arms race between host and phage happening. And to make this work for a food packaging system becomes almost impossible, because you would have to have constantly changing phage cocktails to be attached and delivered to the manufacturers of those materials. And so for this reason, going with simple enzymes has significant advantages. One of them is actually also biotechnological scalability. But there's one more, and that is that lytic enzymes, so endolysins that kill the bacterial cell walls, are very widespread in nature. So we have hundreds, sometimes thousands of different endolysins that can be harnessed for use. So here, for example, we have 3 different endolysins that could be displayed on our bioparticles. And so if we say would use the first enzyme and we would find emerging resistance, all we have to do is create a different gene fusions that takes the second or third enzymes and displays on the bioparticle. And that can be done within a week or 2. And then instead of having the first enzyme displayed, you display the second or third without having to change any of the production or purification or other manufacturing processes. If you compare this, for example, with antibiotics, then you're looking at a turnover cycle of maybe 10 to 20 years to find a new antibiotic, bring it to market, get it approved, get it to scale. Here, we can reduce it from 10 to 20 years to maybe 3 months. So there's a significant advantage of staying ahead in the arms race of bacterial adaptation and resistance building. But there's also another advantage to lytic enzymes, and that is their modular design. So as you can see here, there are lots of individual functional domains within the endolysins, the lytic enzymes. And the US group at the USDA around Donovan in the early to mid 2000s actually has demonstrated that you can take individual domains from different lytic enzymes and, like a Lego system, click them together. So you take different domains, put them together, change the orientation, change the number, very much like synthetic biology. And then he looked at how active are those? And if you can see here, it describes the fold change in minimal inhibitor concentration. You can actually achieve almost 1,000-fold increase in activity and lack of resistance forming by changing and playing with the orientation and order of those functional domains, which means that not only do you have the large variety of naturally occurring lytic enzymes at hand, but you can pre-build libraries, combinatoric libraries of synthetic chimeric endolysins. So when resistances occur, you already have varieties out there that you can test and use those new varieties that become active to put into the production, again, without having to change any of the underlying production purification and the manufacturing processes for the materials. And so for me, this is a very exciting prospect because it is not a one shot. It's not a one trick pony. It's actually a sustainable way of providing antibacterial resistance, antibacterial mechanisms over a long time and being sustainable about it. But nothing is without challenges. And what we have found in our research so far is that if we are dealing with psychophilic cold-loving bacteria such as Clostridia that cause the blown package spoilage, we run into problems or challenges rather in terms of how do we actually quantify the activity of those endolysin displaying bioparticles. And we found that because those Clostridia grow at, say, 4 degrees Celsius, we also slow down the activity of our enzymes. And in our assays, any background contamination has a tendency to take over. So it becomes very difficult to actually fully assess the activity of our functionalized bioparticles. And so the AG research team has started developing new assays that are actually suitable for measuring bactericidal or static activity of our functionalized bioparticles with those cold-loving bacteria. And essentially it goes, it is based around the premise that we don't need to have growing cells present. All we need to have is cells. We can grow our indicator strains in larger quantities without grow them up, harvest them, mix them into buffer, and then add our functionalized bioparticles, where we can then measure optimal density and then quantify the number of cells that is actually immediately destroyed or inhibited by the activity of the endolysins displayed in the bioparticles. And then also do plate counts, because then they can grow, the surviving cells can count and enumerate them. So we're currently optimizing those assays to get a better handle on quantifying the effects that we have on these more difficult to quantify bacteria. Another interesting aspect is also now we're shifting gear a bit, we're going to material sciences, is the surface properties of our films and how that affects how we need to look at attaching and working with those films. So we have 2 different films, A and B. Both are biodegradable films provided by Convex, but they have very different surface properties. So here in film A, we can see with the atomic force microscopy, it's a very, very smooth, even surface. There's hardly any dips and valleys or mountains coming through. And that is confirmed by electron microscopy. So you see here on a 5 micrometer scale, it very much looks like a almost flat film. This is quite in contrast to our film B, where we see that in atomic force microscopy, you suddenly see distinct valleys and hills coming through the surface, and under electron microscopy, you can actually see it much better. So you have those valleys, you have the hills, but you also have ridges in here. And if you think again of the scale here of 5 micrometer, our bioparticles are about a 20th of that size. So they could easily fit, say, into those ridges and valleys and potentially be hidden away from the direct material to food contact, which is required for the endolysins to destroy the bacterial microbial cells. Whereas here in film A, there would be no way the bioparticles could hide away, and all the enzymes that are displayed on them could actually come into direct close contact with the food items. So the type of film becomes very important in terms of suitability for coating with bioparticles. But it surfaces also a different attribute, and that comes about the spraying itself. How do you create even films in coatings onto those films? And again, if you're looking at film B for now, here with the water contact angle, you'll see we have a very distinct bubble forming with a very steep angles. And if you think if you imagine you spray onto the surface, you get little distinct droplets that would then create islands of bioparticles deposits, but not a coherent film. Versus if you have in film A, it's a much rounder droplet with much shallower angles. And again, if you imagine spraying this, the droplets can much easier coat a much larger area and create a smooth, coherent film together, which is much better for an even distribution of those bioparticles. And finally, if we switch over to our edible films, in that case, we're looking here at dissolvable cranberry films. So this you can actually take and eat, and it dissolves in water. And if you apply these types of films, they have inherent biological properties already. In this case, for example, they can provide oxygen barrier functionality. It's quite impressive if you coat, for example, bananas in there. After 10 days, we have our normal brown banana that nobody likes to touch. But if you coat the banana with, say, these edible films, you see that there's hardly any oxidation happening after 10 days. So this would be very much still usable and attractive for any consumer to eat. The same, for example, with green beans. Without treatment and coating, after 15 days, they look very shriveled and unappetizing. Whereas if those are coated, they pretty much look very usable and edible still. And again, because we're looking at dissolvable cranberry films, if those beans would be cooked, the film would dissolve within the water or steam, and you would not actually have any detrimental organoleptic properties left. And with that, I am slowly but surely wrapping up. One of the things I would really like to go into is diversification in the future. So that's part of what the center is about. What can we do beyond smart bioplastics? One thing for sure is that we need to look at resource streams for bioparticle production. At the moment, of course, for lab scale, we're using defined media, and they are not really suitable for large scale manufacturing because they're too expensive. But can we use other resource stream, low value resource streams like forestry, agriculture, or wastewater, say here lignocelluloses from wood, animal manure, food waste, industrial residues, or sewage sludge, to then create new bacterial production strains that are optimized to break down and utilize the carbon and nutrient sources provided in those low value food resource streams, and actually create some and manufacture the bioparticles in a much more sustainable and affordable way. One thing that seems a bit counterintuitive, but has brought a lot of industry demands for industry reference group, is to explore the option to actually use our functionalized bioparticles on petroleum-based plastics. And again, this is something we could see as an intermediate step to introduce the whole technology platform into the food packaging market. and at least help reducing the overall complexity of the layered food packaging materials itself. But this would certainly be, while it is industrial commercially very desirable, because we can use a large range of existing films, we would only see this as a intermediate step to buy us possibly a bit more time to get the biodegradable and compostable films on scale and economically viable. But then we're also looking at completely new functionalities that go way beyond smart bioplastics. For example, we could look at bioremediation, we could use at health, we can use at food applications, but we could also use, and I'll just have this here in looking here, for example, biofilms in meat, fish, or dairy production plants, where surfaces, metal surfaces and pipings are often inaccessible for thorough cleaning and provide ample ground for bacterial contamination and then biofilm formation. And it is very hard to get rid of those biofilms, even with harsh chemical treatments that are very aggressive to the environment and to the workers that have to apply them. And if we could use functionless bioparticles, for example, that degrade those biofilms and then kill the bacteria that are harbored and protected in there, you could actually have a sustainable, non-toxic way of keeping production facility, food processing plants compliant with and reduce the level of bacterial contamination. And with that, I just want to introduce you quickly to our industry reference groups. We currently have 9 members going from small, medium enterprises to very large consortia in New Zealand. We are actively looking to increase our industry reference group. And if anybody is in the audience is interested in joining, please get in touch with us either by email or message us on the center website, and we can get in touch. And nothing of this really is done by myself. We have a rather large team currently in here. Messe University with Nigel French, Ryan and Neroli, who are running the lab. They're currently in the process of hiring not 1 but 2 PhD students and the summer interns. They will hopefully come this calendar year all. Our friends at Scion, led by Robert Abel, do all the material science and existing bioplastic films. Oregon State University is looking at the edible food packaging and coatings. Ag Research, led by Gail Brightwell and Stuart Ledgard, is looking both at the bioactivity against food spoilage of our bioparticles against food spoilage and pathogenic organisms. And Stuart is leading the life cycle analysis. PPDX and Oryx is also based in Oregon and provides custom solutions for specialized bioparticles production. Myself at Blue Barn Life Sciences, so I look with Nigel after the program. Karen Schofield, our program manager, and at North Carolina State University with Professor Bruno Barthena. She's a recognized specialist in industrial fermentations. So we work, the method team works very closely together with him to look at fermentation optimization in bioreactors. And here's the team, and I thank everybody for their attention and looking forward to see if there are any questions. I think they can stop sharing now.
Great, thanks for that, Eric. I always learn something new every time I hear this. I think it's my 3rd time I've had this presentation. But
Poor thing.
We've got one question already. There's quite a lot of questions within the question, so I'll chunk it into 3 separate parts, and then we can, and they should flow on quite nicely for one another. So thank you for this fascinating presentation. Can you speak to the additives needed to ensure the functionality of the end product, such as melt strength enhancers, heat stabilizers, plasticizers, antioxidants, impact modifiers, and fillers? So basically, are they do you have to add anything to the smart bioplastics to make them plasticky?
So the smart bioplastics themselves, no, they are individual particles that are simply surface attached or embedded into the materials. Where we are looking at things like plasticizers, and again, those should be sustainable and food grade, is particularly with the edible coating and the edible materials at the moment, to make sure that those materials, when they're brought together with the smart bioplastic particles, still have the right consistencies. They are not too brittle, they are stretchy, they have the barrier functions. And so these attributes still need to exist, and that's where you bring in your plasticizers and other additives.
Great, thanks. And the second part of that question is, are all of these smart bioplastic products intended for both composting and recycling? Do you have a team working on ensuring these material flows can be fully circularized in a range of waste infrastructures to avoid methane releases or waste stream contamination?
Yep. And again, so the answer is two-part. The bioparticles themselves are fully biodegradable. They are naturally occurring storage vehicles, so they are already there in nature, widespread. So there are systems that build them up and systems that break them down. And if they're broken down into butyrate, which is a short chain fatty acid, and that goes straight back into the central energy metabolism, and amino acids. So they are, within 3 months, there's nothing left of them if they're composted in the field. All the actual plastic materials, the packaging materials, it is our aim to use materials that are either biodegradable, which is sort of like the least favorite one, because often you need specialized facilities for biodegradation. Much more favorable is home compostable materials, so that's our second, or the edible ones, which is our most favorite solution, because if they're edible, they are fully digestible. They don't leave any biodegradation, any bioresidues. They can be recycled. They can be put back into other fermentation processes if you're looking about circularity, without leaving any residues. So that would certainly be our most favorable one.
So if they're recycled, they don't need to go, they can just be lumped in with normal household recycling waste. They don't need to go to any specialized.
Yep, that's the idea. Yep. So you essentially could even put them into your home compost, and then they contribute to make very nice soil. So after 3 months, they're completely gone.
Yeah, they because I know that there's some issues with home composting systems not getting to the same level, like same temperature levels as the industrial ones. Is that
yes?
Is
so
that
so that's essentially the differentiation between biodegradable and compostable and home compostable. So you can biodegrade, but often you need specialized facilities. Whereas home compostable means they're actually breaking down in your home compost pile.
Right, that's good. And then the final part of that question is, are you planning to use these materials as a layer in conventional fossil fuel packaging? And if so, what is the end of life management for these multi-layer products?
Yes, so this is actually an interesting question, because it has been indicated by our industry reference group that they would very much like to have a petroleum-based package, so a traditional packaging material that is functionalized with the bioparticles. And again, from a environmental aspect, that's certainly the least desirable outcome, because we're adding functionality to a bad product. No, it's not a bad product, but it's one that is not sustainable anymore. Less desirable. But it is currently one that industry wants or needs, and so we will try and see, can we develop something like this, but with the view that it would only be a very defined intermediary step, and it wouldn't be the primary outcome for us. But on the other hand, the program is there to deliver something for New Zealand industry. And if that is identified as an industry need, then the money we got from the government would be well spent developing something that's actually useful. Again, with the view that it is not the final outcome. And in terms of life cycle, unfortunately, then while the bioparticles on those materials would again degrade very nicely, the end of life for those would not change to the normal non-functionalized varieties.
Thanks. I have another question here, and I'm just going to tag it onto something else that I was going to ask. They said, sorry if I was not listening well enough, but can you confirm the time frame for the project? And then I also wanted to ask what the next steps were after that with regards to commercialization.
Yeah. And so the program is an MB Endeavor program, which runs until September 2026, whereas the center has been set up to ensure that the research and commercialization goes well beyond the end of life or end of the in the MB Endeavor program. So the next steps in commercialization is certainly the upscaling. So that's where we're working very hard to create these stable production strains to make this possible. We're looking at national, international partners that actually have the infrastructure for upscaling as well, and purification. And we are also trying to get hold of New Zealand, Australian regulatory authorities like FSSRC and MPI to explore what is needed to get those functionalized bioparticles approved either as a food additive or as a food packaging additive.
Great, thanks. I've just had another question come in, which I think might have been addressed, but are there any shelf life enhancement capabilities of these bioplastics?
Yes, so there are bioplastic materials already that can be functionalized, say with essential oils, modified atmosphere packaging. I think silver particles, lysine is being used a lot. The problem there again is these are often, particularly if you take lysine, is a one-trick pony. Lysine has been used extensively as a agent to inhibit bacterial growth, very successfully so, but in recent years, resistance has started cropping up, which means that lysine becomes less and less effective. Problem is there really isn't anything else there that could replace lysine as effectively as lysine does. And so this is where the bioparticles, the endolysins, come in, because they can be rapidly changed. And so as bacterial resistance emerges, we can actually simply change the endolysins displayed on the bioparticle and overcome the resistance immediately. So I think that is perhaps the most attractive feature.
Great, thank you. If there's any more questions, be quick. No, I think we're good. In that case, then I'll close off our session. Thank you, Eric, for your time and expertise and sharing your work with us. This will be available on the NZFSSRC website shortly, and you can use that to send to any of your colleagues or people that you're trying to educate more about this as well. So thank you, Michael, for putting that online. And as I mentioned earlier, the videos from our symposium on the 3rd of July will also be there in the next couple of weeks. Our next seminar session is with food safety culture guru Frankie Annis on Wednesday, the 30th of August at 12 p.m. So watch your inboxes for the registration details for that. So in the meantime, thank you for your attendance and have a good rest of your day, and we'll see you next time. Tēnā koutou katoa.
Thank you.